Welcome to another video lecture on topic D2.1 Cell and Nuclear Division. This is our additional higher level content. So my SL friends, you are not required to listen to this video. You will not be assessed against this content, but it's pretty interesting, so definitely feel free to keep listening if you would like. Our guiding questions continue to be: how can large numbers of genetically identical cells be produced, and how do eukaryotes produce genetically varied cells that can develop into gametes? We're doing quite a bit less of meiosis today. We are focusing mostly on mitosis and those genetically identical cells. Our objectives: we're going to describe the rules of cell proliferation, collecting lots and lots more cells. That's mitosis. We're going to also outline the cell cycle phases, so not just mitosis, but the whole entire cell cycle. We're going to describe the roles of cyclins and cyclin-dependent kinases, or CDKs. We're going to talk about the effects of mutations in proto-oncogenes and tumor suppressor genes. You can probably guess that's going to lead to the formation of cancerous tumors. Those tumors can be benign or malignant. New cells are generated by cell division, mitosis and meiosis. We're going to talk a lot about this proliferation, or increasing the numbers of cells that we have, as an important factor in being alive. Plants, of course, are going to grow new cells via mitosis, cell proliferation. Plants have some super cool tissues called meristematic tissues. This is basically the stem cells of plants. Plants have a couple different kinds of meristematic tissues. We're going to talk just about two today. There is apical and there is lateral. So apical meristem is at the shoot and in the root, and it's all about lengthening the plant. So the plant can get taller, the plant can grow deeper roots with this apical meristem, so roots and shoots. There's also lateral meristem around the stems, the branches, and that increases the width of those branches. When we're looking at tree rings, we're looking a lot at how that lateral meristem has increased the width of the tree trunks. We animals also go through lots of mitosis to grow additional cells. We start as one super cool fertilized egg known as a zygote. That zygote's going to go through mitosis. We're going to proliferate or increase our numbers of cells via mitosis again. So we go from 1 to 2 to 4 to 8. Eventually we have this ball, this solid ball of cells, like hundreds of cells at this point, known as a morula. These cells then migrate to the outside of this sphere, become a hollow ball of cells known as a blastocyst. Of course, just mitosis is not sufficient for us to develop into the amazing creatures that we are. There also needs to be some differentiation. And so what happens after this blastocyst stage, we have this super cool thing called gastrulation, and then we get these dents, these invaginations start to form in this ball of cells, and that allows for these cells to become different germlines, and we end up with spinal cords and brains and hearts and muscles and digestive tracts. So pretty cool, the differentiation piece, of course, but none of it is going to be very convenient for us to develop into humans until we've got all that mitosis going, so that we have a giant ball of cells that can then turn into us. This proliferation of cells via mitosis is not just for growth. We also use it to replace cells and to repair tissues. Here we are showing a little diagram of some cells, a crypt in an intestine. And what happens is we've got these super cool stem cells. These stem cells here are going to divide via mitosis. And then those cells, as they divide, they're going to grow up and then push out of this structure known as a crypt. Because what happens is all the food or feces or whatever kind of intestine we're looking at here, as it is pushing through the colon, we end up with a lot of damaged cells. So we're killing these guys off pretty quickly. We replace the entirety of our intestinal lining every 5 to 7 days. That's how fast these cells die off and how quickly these stem cells need to go through mitosis to push these cells up to replace the ones that are dying. Be super careful. We do replace cells via mitosis. We do not repair cells. That would imply that we're taking some cells and we're fixing them. We're not actually repairing the cells. We are replacing the cells. We talk about repairing tissues, replacing cells. Be careful, we cannot quite use those interchangeably. In our first lecture on cells and nuclear division, we briefly mentioned the cell cycle, though we talked mostly about mitosis. But there are these other super cool stages of the cell cycle. We've got G1, which is our first gap, but we're going to think about how we've got some growth going on. So G for gap, but also G for growth. We have G1, that first gap, G2, that second gap. Here is where we are going to replicate our mitochondria. If we're plants, we're also going to replicate our chloroplasts. During the S phase, we are going to synthesize some DNA. This is our stage of DNA replication, which is incredibly important because if we didn't copy the DNA before we divided the cell, then we would have some cells that were anucleate or without a nucleus, and that not super functional. So again, we've got G1 and S and G2. These are all the pieces of interphase. The cell can go into mitosis after that. We've got prophase, metaphase, anaphase, and telophase, those pieces and parts of mitosis, and then cytokinesis, the cell division. Mitosis is all about nuclear division. Sometimes, especially our very differentiated or specialized cells, will exit the cell cycle, and instead of continuing through G1, S, G2, and back to mitosis, they will instead enter mitotic arrest, which just means we're going to stop mitosis, and they will not divide. Instead, they just do their thing and then die. The cell cycle is controlled by these super cool proteins called cyclins. We have lots of different kinds of cyclins, and the levels of these cyclin proteins will raise and lower. This concentration will go up and then go down at different phases of the cell cycle. So again, here's our G1, that first gap, S, synthesis when we copy our DNA, G2, that second gap, and then mitosis. What's pretty fabulous, if we don't get just the right levels of these proteins, then we do not move into the next stage of the cell cycle. And this cell instead would go to that G0 or mitotic arrest. And the reason is if we're not making the right levels of these cyclins, that means there's something wrong with the cell, and we definitely do not want to make more copies of broken cells. So these cyclins help to regulate the cell cycle, make sure that we are copying cells that are healthy enough to make the right levels of cyclins, which again just keeps us proliferating the good cells and not the broken cells. This is quite a bit more detail here than we need to know about cyclins, but it's an interesting look at how these cyclins work. So these cyclins, these proteins, are produced by genes. Of course, we need the genes to be activated appropriately to make the right levels of cyclins. These cyclins will then bind with these CDK molecules. These are cyclin-dependent kinases. And their job is to bind with the cyclin to then form a functional enzyme. So these two guys work together to function as enzymes to carry out some function that is needed to move into the next stage of the cell cycle. If there isn't enough cyclin produced, then there aren't enough of those complexes, which means that the enzyme activity is low, which means that you're just not going to move to that next stage of the cell cycle. Super interesting. Of course, we're going to regulate also the cyclins and the CDKs. All of these are references to some inhibitors that are going to slow down the rate of these enzyme complexes, the cyclins plus their CDKs, their cyclin-dependent kinases. Cyclins are just one mechanism of cell division control. We're going to look here at two more genes that have an impact on cell cycle, the regulation of the cell cycle. The first one that we're going to look at is a proto-oncogene. These guys most of the time are going to code for something like a growth factor, so a protein that helps to promote cell division. If we have a normal functioning proto-oncogene, then we're going to promote normal amounts of cell division. If, however, we have a proto-oncogene that is mutated, then instead of calling it a proto-oncogene, it's now just an oncogene. That prefix onco, oncologists, this is all about cancer, and most of the time when we have cell division gone awry, it is going to lead to this disorder that we call cancer. When this oncogene is mutated, we're going to have too much of that growth factor being produced. Too much growth factor means too much cell division, and we end up with a mass of cells from all of that extra division. That mass is known as a tumor. And again, this is a symptom, a consequence of cancer. An example of a proto-oncogene is the HER2 receptor gene. So HER2 stands for human epidermal growth factor receptor number 2. And when we have normal amounts of these growth factor receptors, then our cells are going to divide at a normal rate. If, however, we have a mutation that we're going to call now this positive, so HER2 positive cells have a mutation that leads to too many copies of the gene, which leads to too many copies of these protein receptors, which is going to lead to far too much cell division. We have lots of medicines that are specifically related to HER2 positive cancers, especially breast cancers now. But historically, cancers that were HER2 positive were known to be very aggressive and really hard to treat. At the opposite end of the functioning spectrum from those proto-oncogenes are the tumor suppressor genes. And you probably guessed that their job is to suppress the formation of tumors. So an example of a tumor suppressor gene that we talk about quite a bit is P53. So what happens with P53, if there's some kind of environmental stressor being put on our bodies, then P53 is going to send our cell into mitotic or cell cycle arrest. It could be that there is some cancer growing and we don't want to grow more of it, and so P53 shuts it down. Or there could be some DNA damage and we don't want to copy cells that have broken DNA. That's bad. It could be that we are very hungry. If we are starving to death, it doesn't make sense for us to make new cells because there's no food to support those new cells. There could be a hormonal thing going on, some other physiological process that would make it more beneficial for us to not be making lots more cells. And so P53 takes this information in and then sends a cell into mitotic or cell cycle arrest. It could be that that cell is just going to hang out. Senescence is just do your thing, kind of go dormant for a little bit. Could be that we're going to turn on some DNA repair mechanisms to fix whatever damage was happening. Or it could be that we just need to get rid of that cell completely. So we could have some form of pre-programmed cell death like apoptosis or autophagy. This is where our white blood cells eat our own cells because they're broken and we don't want to make more copies of them. Whatever it is, P53 is going to shut down cell growth of a cell that should not be growing, shouldn't be dividing. If we mutate this P53 gene or one of our other tumor suppressor genes, if we mutate it, then we don't have that stop mechanism in there anymore. And we can have that DNA damage, and then we are not preventing those tumors from forming. And then we end up with those cancer cells proliferating. We have these masses, these tumors, and that's bad. So we definitely want to keep our proto-oncogenes proto and not onco. We also want to keep our tumor suppressor genes normal. We don't want them mutated either. A common analogy to describe this balance that leads to regulation of the cell cycle is with this idea of a race car. And it's a really nice analogy, which is why so many of us like to use it. So in a normal functioning cell cycle, what we have is a little bit of proto-oncogenes turning on some gas, pushing on that gas pedal, saying, let's go ahead and make some cells. But then the tumor suppressor gene is like, ooh, but not too many. And proto-oncogene is just a little bit. And tumor suppressors are like, okay, but just a little bit. When we have some mutations, if the mutation is in the proto-oncogene, now we have too much gas and we're like, let's go, let's go, let's go, and we're making too many cells. If we have a mutation in that tumor suppressor gene, then we don't have a break anymore. There's nothing to slow down those proto-oncogenes and oncogenes, and so again, we end up with just too much cell division, and then we have those tumors that are symptomatic of cancers. We can actually look at the rate of cell division to help us diagnose and predict cancers. We call this counting cells that are in mitosis the mitotic index. mitotic index. This is just us looking at the number of cells that are dividing, the number of cells that are actively in mitosis divided by the total number of cells in our sample. So here we're looking at some data from a study that was done on a chemical that can help to slow down cell division. So this is an inhibitor of mitosis, and we can see that these treated doses, the bigger the dose and the more the amount of time, the lower this percentage of cells in mitosis is compared to what our control was. So we had 1,741 dividing cells, and that was equal to 36.37% of all the cells, which means I did some backwards math. We must have had 4,787 total cells. That 1,741 of them that are in mitosis, do that division, multiply by 100 to turn it into that percent. I definitely scribbled on the percent sign. There it is. So there's our percent sign. And so 1,741 total cells that are dividing actively in mitosis, whether that's prophase, metaphase, anaphase, telophase, or cytokinesis, all of these would be considered actively dividing, divided by the total number of cells in the sample, and that gave us our 36.37% of the cells are dividing. The higher the mitotic index, the greater the number of cells in mitosis, the more likely that that tissue sample is cancerous. So those mutations in our proto-oncogenes that lead them to oncogenes or that break our tumor suppressor genes, those mutations are caused by things that are called mutagens that literally generate mutations. Lots of different mutations can occur, lots of different causes for those mutations in our cell regulatory genes. Some of them are inherited. Sometimes we are just born with genes that make us more susceptible to cancers, but then there are some environmental mutagens as well. Radiation is one, which is super interesting because radiation can cause cancer, but then we also use radiation to kill cancer. UV rays from sunlight, please my friends, sunscreen every single day. Overusing X-rays can also lead to some radiation impacting our DNA health. Carcinogens in things like cigarettes. So carcinogens are going to be chemicals that can lead to cancers. All the junk that's in cigarettes, it's so bad for us. And so are e-cigarettes, my friends. Vaping is bad for you. Don't do it. Lots of junk in our food, some processed foods, some preservatives in our foods, cosmetics, cleaning products might also lead to some mutations. And then also we can get sick. And sometimes these bacteria, these viruses, like human papillomavirus, can cause cancers. HPV is very commonly associated with cervical cancer. And again, these tumors are the consequences, the symptoms of cancers. We're going to talk about two kinds of tumors, primary tumors and secondary tumors. Primary tumors are known as benign because even though they're not good, Benny means good, they're not as bad as they could be. Our secondary tumors are much worse, and we call those malignant because mal means bad. So what's happened here, we have our mutated cell. So either the proto-oncogene is now an oncogene or our tumor suppressor gene has gone awry. Something's happening and this cell is dividing far too rapidly and we end up with this primary tumor that is benign. What is good for us humans is that doctors are pretty good at surgically removing these primary tumors. So we just cut all of those cells out of the body, throw them away, and we are now cancer-free. That's all the treatment that we need. That's great. And so finding those cancers, finding those tumors early is super important for a positive outcome. If, however, that tumor continues to grow and eventually detaches, we have some cells detach from that primary tumor, get into the bloodstream and travel through the body to some other tissue type where that cancer cell can then start to invade and grow a secondary tumor, that malignant tumor, someplace else in the body. This is when the cancer is spreading. We call this metastasis, and this is bad. So metastatic cancer means that the cancer cells that were here are now in other parts of the body. And it's very uncommon for there to be only one secondary tumor. Most of the time, those cancer cells are spreading in lots of places and starting lots of tumors, and it's really hard for us to cut them all out or to treat them all with radiation and chemotherapy. So ideally, we're finding tumors at that primary stage and not after metastasis has occurred. So benign tumors, they're not good, even though Benny would imply good, they're just not bad. Whereas those malignant tumors, the ones that have spread due to metastasis, those secondary tumors, these are super bad and really hard for us to treat. And my friends, objectives are accomplished. We did it. We talked about the rules of cell proliferation. That was lots of growth. In plants, meristematic tissue are the stem cells. We also have some cell replacement and tissue repair happening via mitosis and that cell proliferation. Again, be careful. Don't flip those replacements and repairs. We don't repair the cells. We replace the cells to repair the tissue. We talked about our cell cycle phases. We have interphase, which is composed of gap1 and synthesis and gap2. G for gap, not for growth, but remember G for growth as well, and then synthesis, DNA replication. We have mitosis, which is prophase, metaphase, anaphase, and telophase. We have cytokinesis. Mitosis is all about dividing the nucleus. Cytokinesis, we're dividing the rest of the cell. Sometimes our cells will go into G0 or mitotic arrest if we do not want to make more copies of them. We talked about the rules of cyclins and CDKs, cyclin-dependent kinases, in terms of regulating the cell cycle. If we don't have the correct numbers, levels, concentrations of cyclins, then we don't move into that next stage of the cell cycle. We instead head to mitotic arrest. We talked about proto-oncogenes and tumor suppressor genes as being very important for regulating the cell cycle, and if those guys are mutated or broken, then sometimes our cell cycle goes awry, and that is known as cancer. A tumor is a consequence of cancer, and they can be benign, which means that they're not good, but they're not really bad, or they can be malignant, or they're really, really bad. And that's it. Good work today.